Hello and thank you so much for joining me. Today I'm going to do another presentation on CRISPR Cas9 gene editing for T-cell therapies because I feel that I didn't cover some things in great depth in the presentation that I gave previously. So this is going to be the content. It's not going to be a very long presentation. It's going to be on HER2 or HER2 targeting CAR T cell therapies because normally, or at least the ones that I've done in the past, has been CD19 or B cell mature antigens. So this particular therapy is in development. It's not yet been marketed. It's still in clinical trials. Where the target antigen is this HER2. So I'll be talking about that. And then I'll be talking about multiplex editing and Cas9. And the reason is because I did mention it in my previous presentation, but I didn't really talk about it in that particular presentation in depth, or at least you can end up going down a rabbit hole for these things. So I feel I kind of glossed over it. So I just think that I just wanted to clarify a few points. And then I also want to talk about these synthetic genes again, because I did gloss over it in the last presentation. I guess you can think of the last presentation as an introduction. And then the last thing I wanted to go through is these steps in these induced pluripotent stem cell engineering, how we manufacture the products, or at least I'm not going to go through exactly all the steps with regards to the manufacture of the product, but just the critical things and the critical points, because perhaps I didn't emphasize it, so I just want to emphasize it now. So HER2 stands for human epidermal growth factor receptor 2. So it's a receptor, a transmembrane receptor, tyrosine kinase, and it's encoded by the ERBB2 gene. And it's normally involved in cell growth and differentiation signaling pathways. And the reason why we are interested in it is because it's overexpressed in many cancers. So in breast cancer, up to 25% of cases, in ovarian cancer, gastric cancer, lung cancer, and colorectal cancer. And this is the reason why it's been selected as a potential target. However, there are some issues with it, but we have ways of overcoming them. So, as I said, it's overexpressed in tumor growth, and also we have where there is poor prognosis. It's already a successful therapeutic target for monoclonal antibodies or antibody drug conjugates. So it's something that's already has a track record. And the reason that we're sort of exploiting it or exploring it for CAR T is because of the fact that we want to target solid tumors. However, as I said in the previous slide, there are some challenges with on-target, off-tumor toxicities. Yeah, so we want to have a way of tightening the control to ensure that we have better safety for these types of targets, or these types of CAR T where we're targeting HER2. And the way we could do that is, I've listed it there, low affinity CARs, dual antigen targeting, suicide switches, and tuning of CAR expressions. I'm not going to go through every single one because that's a presentation in itself, in my opinion, but like tuning your CAR expression or tuning of your affinity of your CAR. So high affinity CARs may bind even to low levels of HERs on normal tissues. Yeah, so some tissues have this low level HER2 expressed on them, and we don't want to have high affinity chimeric antigen receptors that will target those tissues, because those tissues will be healthy, and we don't want to have that toxicity. So if we use low or moderate affinity, and that's using single chain variable fragments to preferentially bind to HER2 overexpressing on tumor cells, whilst at the same time ignoring the healthy cells with a baseline of HER2, then hopefully the strategy's already been accepted, at least in the United States, I think, and actually for other products as well. Then we can have a better way of targeting the tumor cells without targeting healthy cells. And then there's the other possibility, and this is this dual antigen targeting, where we have this AND gate logic. So instead of activating one signal like the HER2 alone, we use a combination of targeting antigens. So it requires two antigens, so HER2 plus some other tumor marker, and only if both are present in CAR T cells do you get activation. And then there are other things, other sort of design mechanisms that we can put into place, like suicide switches, and to restrict the CAR expression only to the tumor site. So those are the sort of, I don't know, I would say not restrictions, but cautious and safety measures that we can take in order to limit the effect of on-target, off-tumor toxicity. So I want to talk about multiplex editing, because I didn't really talk about it for too long in the last presentation. So I put all my notes on this slide because I think it's useful. So multiplex gene editing refers to the simultaneous modification of multiple genetic targets within a single cell using tools like CRISPR Cas9. So basically, you have one CRISPR Cas9 and you have multiple guide RNAs that can target multiple genes and therefore edit multiple genes. So this technique, it allows for a coordinated either knocking in or knocking out or regulation of several genes at once. So you can do multiple or at least complex reprogramming. You can make multiple changes to the gene, and therefore you can make complex reprogramming of the cell itself. And in cell therapy, multiplex editing is used to enhance your therapeutic function. So you would knock out certain inhibitory genes, reduce immune rejection. So that means knocking out your HLA genes, so the genes that that can cause rejection, if you like, of your product, especially if it is an off-the-shelf like product, and you want to prevent graft versus host disease. So you would knock out your native endogenous TCRs, T cell receptors, and you also want to introduce synthetic functions. So you might want to introduce other receptors as well as your chimeric antigen receptors, and those other synthetic receptors are what I refer to in later slides, that will enhance your T cells to enable them to really target those tumors and really have a stronger efficacious effect. And what have I got here? That this strategy is essential for creating next generation allogeneic cell therapies with improved safety, persistence, and anti-tumor efficacy. And I'll go through that in the next slide. Oh yeah, I just wanted to give a list. I don't want to go through every single one here, but there are other tools that you can use for gene editing, multiplex gene editing beyond Cas9. And here they are. So you've got the CRISPR Cas9 itself, and then you've got another one. This is the CRISPR Cas12a, which uses a different bacteria Cas9 system, and I think that this can do. You don't need multiple guide RNAs. You can have it all on the one. However, it does have some issues with it. And then you've got these talons, which is transcription activator like effector nucleases, and zinc finger nucleases, base editors, and prime editors. But the CRISPR Cas9 remains the most sort of versatile, and it's much more widely adopted due to its simplicity and the ability to simultaneously target multiple genes with different guide RNAs. And you have to validate your guide RNA. But once you've done that and you've got confidence in it, you can have multiple guide RNAs to target multiple genes. But each of these systems that I've listed here, they offer trade-off in terms of efficiency, specificity, and design complexity. So here is a cartoon representation of Cas9. The Cas9 enzyme, and it's showing multiple guide RNAs. So the Cas9 system works on a one-to-one basis with a single guide RNA that has one spacer targeting one site on your DNA. So this particular Cas9 protein is from the Streptococcus pyogenes, and it's a programmable DNA endonuclease. So it basically cuts or it breaks DNA. And it does so by introducing a double-stranded break in the DNA using these two active nuclease domains that I've got listed here. So the step and the process is the first step is the Cas9 binds to the guide RNA, and it forms this complex, which is the Cas9 guide RNA complex. The guide RNA itself, it has about approximately 20 nucleotides, and those nucleotides, they're complementary to the target DNA. So the Cas9 guide RNA complex scans the DNA until it finds this PAM site. So this PAM site is the protospacer adjacent motif, and it's essential for recognition. And once it finds that PAM site, it basically locks to the the site, and then Cas9 unwinds the adjacent DNA. The guide RNA base pairs with the complementary target strand. If it's a good match, it will. And then the so I've got here the HNH domain that cuts the target strand. So that's the one with the matching that matches the guide RNA. And then this other domain, the RUVC domain, it cuts the non-targeted strand, which is going to, and it usually cuts it a few bases away, and this creates your double-stranded break with a blunt or staggered ends. If your staggered ends is when it cuts the RUVC domain cuts the non-target strand a few base pairs away, but a blunt cut is where I believe where it's adjacent. So yeah, so I think I've already said this, the RUVC is it cleaves the non-targeted strand, i.e. the strand that's not complementary to the guide RNA. But the other one, the HNH nuclease, it cuts the DNA strand that is complementary to the guide RNA. And the blunt end, if it's a blunt end cut, that means both DNA strands are cut at the same position across the helix, and you don't get any overhang. And this is typical for Streptococcus pyogenes, Cas9. However, if you want staggered ends where the two DNA strands are cut at different positions, creating sort of a short single stranded overhang, you can use a different systems and different variants of Cas9 like for example Cas12a. And that might be better for some homology directed repair applications. So you can use this Cas9 enzyme for not just for making knocking in and knocking out genes, but you can use it for all kinds of things as well. I haven't got into that in this particular presentation, but you can engineer your Cas9 so that one of the nuclease domains is inactivated so that it only cuts one strand. And this might be useful for where you want to make precise edits or base editors. And you can also engineer Cas9 so that both domains are inactivated, so there's no cutting, but it still binds with the DNA. And then you can say, for example, inactivate that particular DNA. Maybe if it's on a gene itself, you can inactivate it, and you can use the CRISPR Cas for inactivating or activating, and for making modifications, epigenetic modifications. So you can do more than just performing multiple edits with CRISPR Cas, but I haven't obviously discussed all of that. So the next stage is talking about these synthetic genes. So remember, we have the gene has been edited with T cells, the chimeric antigen receptors, and we're also including other genes in order to enhance the therapeutic outcome, the therapeutic effect, to target the tumors and to make a better product, and to make a product that can be an off-the-shelf product for all. Right, so these are the genes that, these are the the genes and the products that are going to be sort of knocked in to your gene, your genome. And the reason being is that we want to enhance the actual therapeutic effect. And I did talk about these in the last presentation, but I didn't really go through it in much detail. So TGF-beta chimeric receptor. So what this does, and I'll go through it in more detail in the next slides, is it converts an immunosuppressive environment into an activating one. So the tumor microenvironment, it releases these TGF-beta what's it called? Cytokines, yeah, or chemokines, and that causes, in normal situations, it causes the T cells to be switched off, to be inactive, and basically to become a bystander, which is not what we want, yeah. And this is the effect of the tumor microenvironment. And so what these synthetic genes have done is basically turned it on its head and ensured that whenever this receptor, this TGF-beta chimeric receptor, binds with the TGF-beta molecules, or then it will become activated as opposed to being turned off. And then the CD16A, so this is taking advantage of this is taking advantage of the T cells and using it with other antibodies, other therapeutic antibodies that are used to kill cancers. So basically you get this antibody dependent cellular cytotoxicity because the CD16 is a FC binding site, and it basically enables your CAR T to work with therapeutic antibodies. I've focused this presentation on T cells and not on natural killer cells, but I'll be doing some case studies where I'll be looking at natural killer cells. And then we've got this other gene, and this is this other product, and this is a CXCR receptor 2, and this is basically enabling the T cells to basically home into the tumor cells, to basically find the tumor cells, to traffic them towards the tumor cells themselves so that they can, you know, do their job. So tumor cells are generally sort of cold, and you can't, and not invisible, I think is a good word, and therefore it's a way of allowing the T cells to actually be really target the cancer, the tumor cells, and have that therapeutic effect. And last is the CAR, the chimeric antigen receptor. And this is knocked in, and we're normally knocking it into the T cell receptor alpha constant region. Very rarely in the AVVS1. I'll talk about that, or the CCR5 loci. I'll talk about those loci later on, but just knocking in the actual receptor that will produce the therapeutic result or therapeutic response that we're looking for. Right, so let's talk about these receptors. So the first one that I've got on the list is TGF beta receptor. So this receptor turns a tumor defense mechanism into a weapon, basically. TGF beta stands for transforming growth factor beta, and it's a cytokine, which is a signaling protein that cells use to communicate with one another. But in the context of this tumor biology, this TGF beta receptor is responsible for driving the cells to this T regulatory Treg, which then becomes in effect useless. So it's mostly known as being immunosuppressive, and that's what it's meant to be, because you don't want to have naturally an overreactive or an overactive immune system, because that will cause autoimmunity. And so the body has all these processes in place to prevent that. And this this transforming growth factor beta cytokines. They are, you know, a powerful immunosuppressive, and they're found, and the problem is that, or the challenge is that the tumor microenvironment overexpresses these particular you know, they're overexpressed, and then consequently the T cells are unable to you know, to kill them off and to kill off the tumors, and therefore we get the problems that we have, where the tumor then metastasizes, and then you've got death. And so what the strategy is to reverse that. So instead of deactivation, the TGF beta receptors have been combined with an intracellular signaling domain, and that intracellular domain signals as opposed to suppresses. And so consequently, instead of being switched off, the T cells are switched on. So what have I got here? So so this receptor is created or engineered by fusing two different receptors. So one is the actual receptor itself, and the other one is the CD3 or the CD28. And so in cancer, tumor exploits TGF beta to suppress the T cells and natural killer cells activity, which I've said, to promote regulatory T cell formation, which normally happens, you know, when you have your CD4 plus, with these TGF beta, you get it drives it towards or differentiates towards the Treg formation, but unfortunately it promotes tumor angiogenesis, which is the formation of these sort of arteries and veins, if you like, capillaries within the tumor, and feeds the tumor cells that are growing that you want to avoid. And it means that if you get immune evasion, and also it promotes tumor cell invasion. So it really isn't what we want. And so when you have this TGF1, sorry, this TGF beta that binds to its natural receptor on T cells, it tells the T cells to chill out and, you know, don't attack. But, you know, in the tumor setting, that's a major problem. So, you know, how is this sort of engineered, and how is this set up? So basically you have the extracellular domain, and this is where you have the chimeric receptor. And you have the intracellular domain, and you basically the intracellular domain consists of either the activating CD3 zeta or the co-stimulatory CD28 or 41BB. And there isn't a direct conflict between, because there isn't a direct conflict between the CD3 zeta in this receptor and your normal CAR T, where you have a CD3 zeta in the intracellular domain. So basically you have a complementary, if you like, what's the word, signaling. So you have your normal CAR T, you have your CD3 zeta, and then that's essential for your activation signal. Then you have your co-stimulatory signals that come from your CD28, or it can come from your 41BB. So that's your second signal, and you need to have those two signals in order to get full activation. Yeah. But what this chimeric TGF beta adds is it's number one, it's only activated if you've got TGF beta present, and it then sends a separate signal, and that can be a signal 1 or signal 2 depending on how it's been designed. And basically, if it's been designed with a CD28 domain, you will get a co-stimulation signal. But if it's been fused with a CD3 zeta, then you get activation. And you can get that activation without antigen binding. So these HER2 antigen may not have bound, but you can just get the TGF beta binding to the receptor, to the synthetic receptor, to get that activation. And now what happens is that instead of getting this T cell suppressed, if you like, in the tumor microenvironment, you get activation. So it's the opposite of what normally happens. So basically it allows the T cell to act, and you have that T cell activity in a suppressive tumor microenvironment. But you do have to carefully balance your risk. For example, you don't want to get that hyperactivation, and also you don't want to get off tumor effects, and also you need to think about exhaustion or cytokine release syndrome. So it's kind of like a balancing act, and you might have to consider dosing or having a genetic safety switch, or you know, exhaustion resistance programming, which is another subject in itself. So basically, overall, what I'm saying is that now when TGF beta binds, instead of turning off the T cells, it actually fires it up. So this is just a summary slide, and it's just basically covering, I guess, what I've discussed, and what the TGF beta receptor's role is. And it's really, as I said, it's there to prevent the T cells from not being shut down. Because obviously in the tumor microenvironment, these cytokines like TGF beta, they're everywhere, and they lead to you know, this reduced cytotoxicity. They decrease proliferation, they inhibit certain cytokines, and the thing that I said previously is that they promote Treg development. And in essence, the TGF beta in normal circumstances, it turns the immune system basically into a bystander. And so by hacking, if you like, or hijacking this particular suppressive signal and flipping it, yeah. If the tumor is going to flood the area with this TGF beta, then we'll use it against the tumor. And basically, you're totally redesigning the T cells to remain fully functional, or maybe even enhanced, in environments where normal T cells would be paralyzed. So I'm going to do a number of case studies where I'll talk about various products that are in clinical trials, and talk about what I think the designs are. They'll all be theoretical, but this is kind of a summary, if you like, of what we're looking to. We're looking to get immune activation instead of stronger T cells, and this particular strategy is adopted by one of the products that I believe is in clinical trials. Right, the next synthetic receptor that's been knocked into the theoretical, if you like, case study is this CXCR2, and this is the chemokine CXC motive. Sorry, there should be dashes between that. CXC motive receptor 2. So it's a chemokine receptor, and what it does is it detects signals, and these signals are found in tumor tissues, and basically it's a G protein coupled receptor, and it's expressed on some of the immune cells like neutrophils and certain T cells, and it plays an important role in immune cell trafficking. Basically, it tells the immune cells where to go. Yeah, so this happens naturally with neutrophils and other T cells. And what has happened, what the approach is to design the T cells so that they have that characteristic, so that they can now track the immune tumor microenvironment, which is a major problem. So this receptor, it detects the signals, and then it can then you can then track the tumor microenvironment, and so therefore it has been engineered. So these T cells, not the CAR T cells, but the overall product has been engineered to express this receptor, and it improves their ability to migrate towards the tumors that release this CXCR2 ligand. So basically, the second option is to get the T cells to find the tumor, because the tumor microenvironment tends to make itself invisible. And this is so what we've done is we've exploited how it does that, or we've exploited a characteristic from other neutrophils in order to allow our drug, or the drug, to target the cancer. So this is just an example of the types of chemokines that CXCR2 binds to. And as I said, these are often secreted by tumors, and they're secreted for a number of reasons, mainly to suppress the immune cells, and to promote angiogenesis, and so that improves their oxygenation to, or their nutrition to the cancer cells, and also again to facilitate tumor invasion and metastases. So these chemokines, if you can think of them, they create a gradient, and therefore if we can engineer the T cells in order to follow that gradient, and therefore target the cancer wherever it may go. Yeah. So this is a limitation with CAR T in solid tumors is that there's poor trafficking and poor infiltration, and as I said, that tumor cells, they're often sort of physically or immunologically cold to our T cells. So engineering these CXCR receptors too, it gives that the T cells the ability to detect and migrate towards these chemokine-rich tumors. And so if you combine that with your CAR, your chimeric antigen receptor that is targeted towards HER2, it improves their localization, it improves the retention and effectiveness of the tumors, of the CAR T cells in this inside of the tumors. And the last additional knock-in one is, I think, no, it's not the last one actually, but the one that is in this case study. But I've also included some other knock-ins that may or may not have been used in these case studies. But this is CD16A. So CD16A receptor is an Fc gamma R3A receptor. Now, I talked about this when I was talking about antibodies, and basically they are the receptors that the antibody Fc region binds to, and then they can help kill off the cells that the antibody may be bound to. So if your antibody is bound to, in the variable region, is bound to some antigen, then the T cell, which can be bound to the antibody, can then kill the cell that the antibody is attached to. And basically what it means is that if you're combining this product with monoclonal antibodies, then the T cells will kill off the cancer, even if they're not bound to the cancer, or they haven't bound to the cancer themselves, because they've got this receptor which interacts with the antibody. So it's a multi-pronged strategic approach, where you think of as many different ways as possible of killing off the cancer cell by exploiting the tumor microenvironment, by exploiting other therapies that could be used, as well as the CAR T cells itself, and extending your targeting range of your CAR T cells beyond what they've been engineered specifically to do. Right, so I had here a number of tables. There's going to be a number of tables in this, and this is about looking at what has been knocked out and what has been knocked in, and we're specifically thinking about allogeneic use, because we're looking at an off-the-shelf product. So I'm not really going to talk too much about this because I've already done so in other presentations, but we're looking to knock out your TRAC. This is the T cell receptor alpha constant, and this is the endogenous T cell receptor, and we want to knock in the CAR, the chimeric antigen receptor, in its place. And as we said before, we want to remove endogenous T cell receptors to prevent graft versus host disease, and also to exploit the natural place, the endogenous place where the T cells or receptors should be, and to get consistent expression. And then there's the B2M locus, and this is knocked out. Basically, we want to have an off-the-shelf product, and we don't want major histocompatibility class 1 surface products to be on the surface expression. So in order to avoid your host CD8 T cells from recognizing them. So basically, we don't want HLA A or B or C. We want to knock them out so that the product remains invisible to your recipient, the patient's immune system, and it can act without actually being removed itself. And then we've got this other gene that's removed. This is the CITA, I'm calling it that, CIITA. This basically removes the major histocompatibility class 2 on your CD4 cells. And again, it's for the same reason. You don't want the host to recognize the CD4 plus T cells as foreign or non-self, and you don't want to have what's the word? You want to increase your exposure. You don't want your host to attack this. So this particular gene, it stops when you knock it out. It stops the expression of HLA-DR, DP, and DQ. So these are the major histocompatibility class 2 subtypes. Now you want to knock in HLA-E or HLA-G, and this is because if you knock out the HLA A, B, C, or A, B, or C, if you knock them out, your natural killer cells will attack them because they'll see them as HLA null, and then they'll attack them. So in order to prevent that from happening, you knock in these HLA-Es or HLA-Gs. And then I've got some additional ones here, and this is from my search. In my case studies, I don't think this has been, well, it may have been included, and it may not be information out there for the public. So I've got CD47, and this is knocked in to prevent phagocytosis. You don't always have a number of positions in the genome to knock all these things in. So I don't know what the strategy is that the company took. I've just got this as on my list for the sake of completeness. And then you've got this CISH. So this is a knocked out, and this is CISH acts as a negative regulator for cytokine signaling. So basically, it's critical for T cell proliferation, survival, cytotoxic function, and effector differentiation. So in normal situations, this CISH acts like a brake on your T cell activation. Yeah. So whenever it's induced, it just causes inactivation. It acts like a brake. So when you knock it out, you can enhance your cytokine responsiveness, you can increase proliferation, and you can boost this anti-tumor cytotoxicity. Again, in my case studies, I don't know that they have knocked this out, but this is just included just for the sake of completion. And basically, when you knock it out, you supercharge the T cells, and it just means that your tumor microenvironments are ineffective against the T cells. So, and then last but by no means least, I've got optional here, but this is the PD-1 or other checkpoints. And this PD-1, again, it's to prevent the inhibitory signaling when it binds with the PD-L1 or PD-L2 that naturally occurs as part of the body's defense against autoimmunity. So you knock that out because this is overexpressed. The ligand is overexpressed on the tumor cells. I just want to talk about where we put things because I don't know whether or not I've put this on this slide, but you can, from what I've been able to locate, there's certain places where you can put certain genes. So the track locus, you would put the chimeric antigen receptor or the T cell receptor, or whatever synthetic receptor that you've come together, you're put in for your product. So you knock it out first, and then you knock in the gene that you are interested in, and you have strong promoters. You get uniform CAR expression, and you want to delete the native T cell receptor. So that locus, I don't think I've got track or TRBC, the beta constant, which is optional whether or not you knock it out, because it doesn't really affect it's more important to knock out the T cell receptor alpha constant than the T cell receptor beta. And then there's these other sites. So we've got the AAVS1. So this is a well characterized and well understood locus. It's where the wild type adeno-associated viruses integrate into. So it's well known, well characterized, and it's good for stable expression. And so you can use that. It's known as a safe harbor locus, and you can use it for CD47 or CXCR2 or any cytokine receptors. And then we've got another one called CCR5. And you can use this for your TGF beta domains, genes to knock in, and knocking out prevents it can be used for HIV editing. Again, I'm not going to go through that. That's another presentation, which I'm not going to do, but it's a safe knock-in for knocking out, and it's safe for knocking in. And then we have this B2M locus, and this is the beta-2 microglobulin. And basically, this is to remove your HLA gene. So your HLA-A, HLA-B, and HLA-C. And it's the major histocompatibility class 1. So in some cases, you can knock in HLA-E and HLA-G, and that's to avoid the natural killer attack that you could get. Again, this is for when you want your off the shelf drugs. You want to make sure that you don't get rejection. And then you've got this CETA. This is class 2 trans activators, and it's a popular knockout because it's a single switch that controls the expression of your major histocompatibility class 2 gene. So knocking it out prevents your surface major histocompatibility class 2 expression, even in an inflammatory environment. So it's usually one synthetic gene when you knock in to each locus. So that's the standard approach, because each locus has a limited sort of capacity, promoter, coding region, and regulatory elements. They all take up space. So if you insert too much, you can have the risk of epigenetic silencing or misexpression. So each synthetic gene is knocked into a dedicated well characterized locus. And you do that to ensure that you've got this good controlled expression and safety. And I mean, there are potential designs that you can insert multiple genes into a particular locus. I haven't looked into that, but from what I've looked at, it's mostly one gene in a loci. And you have to, select the locus, and you have to balance your expression level with the cell type and the genomic safety. Right, the other thing I wanted to talk about is the induced pluripotent stem cells manufacturing. And this is just going to be some basics because I didn't really talk about it that much. So the challenge in terms of multiplex editing is potentially you have off-target effects. So you do need to carefully design your guide RNA and optimize your delivery methods to ensure that you get all your desired edits. Yeah, so that's just a caveat. But you start with your induced pluripotent stem cells. I forgot to put that you can get these off the shelf now, or you can order them where they've been manufactured to GMP. And then you do your gene editing, that's with your CRISPR Cas9. And you can knock out endogenous genes, which I've already discussed, and you can knock in synthetic genes or synthetic constructs, and you can do multiple edits. So this is this multiplex editing. Then you select your clone. So you want to isolate the clone, screen the clone to ensure that it's got the correct gene edits, that you haven't got any off-target effects, your genomic integrity, and that you're going to get, as much as possible, your desired phenotypic characteristics. So you want to select the best clone for further use, ultimately. So once you've successfully engineered your cell line, you've selected the best, you've edited your T cells to express the desired transgenes, you've knocked out whatever unwanted genes, you've selected the best clone, etc. You can then generate your master cell bank. And this master cell bank is a bank of cells that have been fully edited, fully characterized, and genetically stable. And from your master cell bank, you create your working cell banks. And these are smaller batches, and they're used for manufacturing or expanding for your therapeutic product. So your master cell bank is critical for ensuring future batches of T cells that have the same genetic modifications without having to go through all the time-consuming engineering process repeatedly. And it just means that the speed of engineering is greatly improved once you've got your master cell bank and your your master cell bank laid down. Once they've been laid down and it's all set up and it's all been characterized and validated, you don't need to start from scratch each time. So it's a uniform pool, if you like, of cells that you can then take and either prepare working cell banks from, or you differentiate into your product. So the initial setup, the first round of multiplex editing, takes time, and it requires careful planning, careful design, careful optimization, so that you can get the correct gene editing. And then after that, and after you've established your master cell bank and your working cell bank, you can use these cell banks for a faster production of engineered T cells which will all have the same genetic modifications. Obviously, you have to confirm that as part of your stability exercise, but it just makes subsequent rounds of cell expansion much faster because you don't need to re-engineer the cells each time. So it's efficient. Once your genetic modifications have been confirmed in your master cell bank, expanding the cells for clinical use through your working cell bank is quicker. And as long as your genetic stability is maintained and you've got data to show that, it's just a much more efficient use and guarantees you're honing down variability. You're preventing that variability from occurring because you're using the same bank of cells. Right, the next stage involves manufacturing, and this will ultimately involve differentiation. So you take your working cell bank, you thaw them, and then you expand it in culture, and then you differentiate down to your T cell lineage. Now, there are two techniques that I've given there, but the point that I want to make is that differentiation itself is, in my opinion, the most critical and sensitive step in this induced pluripotent stem cell derived manufacturing. Batch to batch variability, from what I've seen, is a major concern, especially when you're producing a living drug like T cells. Induced pluripotent stem cells are highly sensitive to microenvironmental cues, to media, to cytokines, cell density, oxygen levels, and passage numbers. So this is really critical any small changes, you can get incomplete or incorrect differentiation. You're gonna get heterogeneous cell populations anyway, but you know, you can, that can vary greatly, and you can reduce the potency or increase your safety risk. So you're not just growing cells, you're engineering cells which has a defined fate, and that has to be consistent and reproducible. Your final product, yeah, again, when you finalize your product, I mean, when you're doing your manufacturing step, this will be standardized. You will have a standard operating procedures, batch manufacturing records that standardize all of these things in order to reduce batch to batch variability. I will talk about that. But then your final step is the product formulation. Your T cells are purified, formulated, cryopreserved, and you perform your quality tests. Yeah. So this is what I really wanted to talk about, because, you know, the biggest critical part and problems occur during, well, I see the challenges is in the differentiation stages. Cells are biological systems, therefore they're inherently variable. As I said, you're going to get lot to lot variations with even your raw materials. Microenvironmental factors can affect how your differentiation, you know, the outcome. And you just need to have very good manufacturing processes, control strategies. And another concept that you need to consider is quality by design, where you build in the quality as part of based upon your understanding of your target product and based upon the understanding of your manufacturing process. So there, particularly during this differentiation stage. Yeah, this is just me giving my opinion here. And so this quality by design approach, it provides a framework, yeah, for your design of your therapeutic product, not just in terms of the safety and efficacy, but in terms of your manufacturability. Especially important for living cell therapies, where batch to batch variability is inherent. Quality by design provides a structure, if you like, for continually assessing, and particularly with process performance qualification activities, you're continually gathering data and assessing and gaining insight and knowledge, deepening that knowledge with regards to the quality with regards to obtaining consistency in quality of your batch of materials. So you continually assess, you continually adapt and refine your manufacturing process based on evolving data. And I've just got you know, what quality by design, I've got a separate presentation for that, but what it involves, which is understanding your target product profile, understanding what you're aiming for in terms of the clinic, what you're aiming for in terms of manufacture, and what you're aiming for in terms of the quality of your product, understanding the critical quality attributes, what needs to be controlled, what attributes have an impact on safety and efficacy in the clinic, and what manufacturing steps have an impact on your critical quality attributes, your critical process parameters and your key process parameters. And you get this information, or you monitor this information from your in-process control data, and you can also get information from your designs of experiments. Understanding what your design space is, having a good control strategy, and a continuous life cycle management in order to you know, have a product that is consistently manufactured under changing and evolving life cycle of that particular product. So it's just a summary slide with regards to induced pluripotent stem cells derived CAR T cell therapy. Basically, the approach, why we take this approach, why we even use it, and that's because of this homology directed repair for editing, but also there are advantages with regards to manufacturing master cell banks and working cell bank. So these induced pluripotent stem cells can be genetically modified or edited, cloned and banked before being expanded and differentiated into T cells. And this process ensures a renewable, consistent and off-the-shelf supply of engineered T cells for cancer immunotherapy. So hopefully that has clarified the gaps that I left in my previous presentation. If it has, then that's great. Thank you so much for listening to this long presentation, which was meant to be short. I hope you've enjoyed it. If you have, please consider giving it a like, please consider subscribing, and please consider leaving a comment. Thank you so much. Bye-bye.